Okay, so DNA structure and replication. Your objective is to be able to explain the double-stranded complementary nature of DNA as is related to the function of the cell. So nucleic acids are a macromolecule, as you guys know. What are the other three types of macromolecules? Pause this if you need to look it up. A macromolecule, biological molecule, they carry our genetic material, that's our DNA. Our DNA contains the genes or the blueprint or instructions for making proteins. Genes are located at certain points on a chromosome. So for example, if the chromosome is the X shape, right? The gene is located on that chromosome. So the chromosome is like the ice cream shop, and the gene is like all the different flavors. The genes are the characteristics that make you, you. Brown hair, uh, uh, red hair, blue eyes, brown eyes. Widow's peak, those are the characteristics that make you, you. And proteins carry out all of our cellular activity. So we have two types of nucleic acids. We have DNA, deoxyribonucleic acid, and we have RNA, ribonucleic acid. The monomer for nucleic acid is a nucleotide. The nucleotide has three parts. They're made up of the sugar. If the sugar is for DNA, it's called deoxyribose. If the sugar is on an RNA molecule, it's called ribose. Remember, sugars end in OSE. They also made up of a phosphate group and a nitrogenous base. The bases for DNA are A, C, G, and T. And the bases for RNA are A, C, G, and U. So DNA is twisted. And because it's twisted like a ladder, we call DNA a double helix. The double helix backbone, the size of the ladder, this is DNA untwisted. The size of the ladder are made up of alternating phosphate and sugars, phosphate sugars. DNA is also known as anti-parallel. It's anti-parallel because one side of DNA goes up, the other side goes down. The bases are like the steps of the ladder. The bases are like the rungs of the ladder. The bases have a weak bond in between them. We call that a hydrogen bond. And the backbone of DNA, the phosphate and sugar, the bonds between them are called covalent bonds. The nitrogen bases only bond to their complementary base pair. So A's only bond to T's and vice versa, and C's only bond to G's and vice versa. Notice how the T is bonded to A, and A is bonded to T, and C is bonded to G, and C is bonded to G. And if you're having trouble remembering that, just remember apple in a tree and car in a garage. I got that off the Amoeba Sisters, of course. So the sequence, this part is really, really important. The sequence of DNA, these bases, they code for a protein. They determine whether you have brown hair, whether you have red hair. Those proteins are key to cell function. That sequence is everything. Please make sure you put a star next to this. There's always a question on the EOC about the sequence of the bases. RNA structure, you saw how DNA is made of two strands. Well, RNA is made of one single strand, And because it's one single strand, the nucleotides, the bases are exposed. And again, the bases for RNA are A to U, U to A. A only bonds to U, U only bonds to A, and C bonds with G, and G bonds with C. So let's try this. If you need to, pause it. So the the bases for DNA are ATCG. You have to know the first letters of all of the bases. The bases for RNA are AUCG. The sugar for DNA is deoxyribose, and the sugar for RNA is ribose. DNA is shaped like a double helix, and RNA is shaped as a single strand. Just some additional information for you. Chromosomes are tightly coiled strands of DNA. Different organisms have different numbers of the chromosomes. We have 23 pairs of 46 chromosomes. You get half of your chromosomes from mom, the other half from dad. Dogs, as another example, have 37 pairs of chromosomes, 74 total. They get half from their mom, mom doggy, mom dog, or 37 from their dad. The genes are pieces of DNA that have the instructions to code for one protein. Genes are pieces of DNA that has instructions to code for one protein. One chromosome can contain thousands of genes linked together. So genes are pieces or sections of DNA, and the chromosomes are long strands of DNA all condensed or bunched up together, like the X. So let's try this. What would the complementary DNA strand be for the following sequence? CGT, ATG. The complementary base would be GCA, TAC. Now I want you to take out your DNA base pairing worksheet and practice on that a little bit.